Well, hello, everyone, and welcome back to Science in the Age of COVID-19. Today, we are extremely excited to welcome Daniela Weiskopf from the La Institute of Immunology here in San Diego. This is my first time hosting the seminar from my new home here at the UC San Diego. So hello, neighbor. We have previously heard from her close collaborator, Alessandro Sette, in the seminar series about the somewhat neglected role of a critical immune player, and that is the T-cell response. Daniela has studied a great deal about T-cell response, activation, MHC repertoire, et cetera, for a variety of diseases and conditions, including dengue fever, Parkinson's disease, and of personal interest to me, environmental allergies, such as grass, dust, and mold. And today she is going to give us an update on the T-cell response to SARS-CoV-2. Daniela.
Thank you very much, neighbor. I'm very excited to have the opportunity to update you on our most recent data on studying the adaptive immune response to SARS-CoV-2. And we are interested in how does this look like after infection and also after vaccination against this virus. So early on in the pandemic, we were interested, does our immune system recognize SARS-CoV-2? And if so, how does the immune system recognize SARS-CoV-2? And this is just basically the three players that we are interested in. So how do antibodies recognize this virus? How do CD4 T cells and how do CD8 T cells recognize this virus? And I'm just going to summarize our initial findings. So early on, we found that measuring T-cell responses against SARS-CoV-2 in people that have recovered from this disease, not only the spike protein, which is the protein that was chosen to develop in a lot of different vaccine constructs, but also a lot of other proteins of this virus are recognized by CD4 and also CD8 T cells. We then thought it's very important that we actually can measure also the T cell response, not only the antibody response against this virus, because it is interesting to understand COVID-19 also for vaccine development. We then developed epitope pools and detected CD4 and CD8 T cells in 100% of the convalescent COVID-19 patients and 70% for CD8 T cells we identified in this samples. We, as I mentioned, found that the T cell response are not only focused on spike, but also on the membrane, nucleocapsid, and other ORFs. And what was very peculiar at this time point is that we also found that SARS-CoV-2 epitope pools were detected in unexposed donors. And I'm going to go back to that in a little bit later. So after we identified that there's a lot of different pools that are recognized, we were then interested in screening that in a much larger cohort. So we have screened that in close to 199 convalescent donors, where we identified that, as I mentioned, spike is highly recognized, but also nucleocapsid membrane and NSP3 for CD4 T cells, and similar proteins for the CD8 T cell responses. What I like to point out at this point is that each donor recognized a number of different antigens. So for example, if you look here on the right side of the graph, there is on average 3 to 4 proteins recognized for CD4 T cells and 2 to 3 for CD8 T cells. And why I'm pointing this out and why I think this is important is what is in the news right now and what everybody's concerned are variants. And if you appreciate that the T cells don't have everything on one target, but recognize multiple proteins, it would take a lot of mutation to completely evade T cell responses. So we saw a pretty broad response. Overall, we identified 280 different CD4 T cell epitopes and more than 500 CD8 T cell epitopes. And not only are each donor is recognizing multiple different proteins, but on average 15 to 20 epitopes. So as I pointed out, this is important when we think about variants, because it would take a lot of mutations in a lot of different epitopes to completely evade the T cell responses. If you look into more detail, if there is any shared epitopes between donors, this is like the breakdown. So you can see that 45% are actually shared in 3 or more donors. So these are promiscuous. That means they are restricted by multiple different HLA phenotypes, and the most dominant are shared between different donors. So there is, of course, also like epitopes that are found only once that are unique for each donors. But overall, a good almost half of the response was like shared between dominant promiscuous epitopes. The other thing that allows you to do it when you know the exact epitopes that are recognized for each of these proteins, you can also draw maps for each of the proteins and draw like immunodominant regions that are highly recognized. And I'm going to show here the spike protein as an example because it is a highly recognized protein in natural infection, but also it's the target of almost all of the vaccines that are developed right now. And if you look at the upper part, which is the blue line, that is the CD4 T-cell responses, you can see that actually the RBD region is not so much recognized for CD4 T cells, but the outside flanking regions are highly recognized for CD4 T cell responses. That looks different for CD8 T cell responses, which recognize the entire spike protein, including the RBD region. And we can draw the same graphs for like other proteins, as you can see here. So to conclude this natural infection, like what are the antigen and epitopes that are targeted by COVID-19? I just showed you that there's a broad repertoire. There's like more than 280 CD4, more than 500 CD8 epitopes. A lot of different HLEs have different repertoire sizes and also recognized it with different strengths. We didn't go into much detail into that. For CD4 T cells, the majority of epitopes are promiscuous, and importantly, each individual recognizes a broad repertoire of T cell epitopes. As I pointed out, there's clear immunodominant regions for CD4 T cell responses, RBDs, epitope poor, but outside of this is the dominant encoding for dominant sites. And the epitope pools that we now have developed facilitate the accurate measurement of all these T cell responses. So now we can actually go and ask the specific question like about all experimentally defined epitopes, like how do they look in different maybe disease severities or also like in the context of vaccination. The next question we had after we identified like what are the targets, how long does this immune response last? So we have collected samples from donors up to 6 to 8 months after infection, again in almost 200 subjects, and you can see here all the different parameters that have been measured, which is on the upper part, antibody parameters. So you can see here spike IgG, neutralizing titers, and also spike IgA. Cellular compartments on the right side here, B cells, CD8 T cells, and CD4 T cells. And you could see that all the way out to 6 to 8 months after infection, you can still detect a very large fraction of T cell responses. And in the case of B cell responses, this actually went up. So there were more responses detectable 6 to 8 months after infection when you look into terms of the 6 to 8 months. So overall, this was the largest study for any kind of viral infection that has ever been done that looked at to like all the cellular compartments, CD4, CD8, B cells, and also in all of the different antibody compartments. So the other thing that you can appreciate from this study is like all of these compartments have different kinetics. So B cells go up and CD4 and CD8 T cell responses have a very different half-life, antibody titers almost plateau. And the last thing that I want to point out here is you can also appreciate the bright heterogeneity of these donors. So you do not know, not all of the donors are still able to like show like immune response 6 to 8 months. So about 10% have undetectable immune response, and there is no hint if you look early on like who will be these 10% that then not able to have a detectable immune response. So there is also pretty wide confidence intervals because there's just like this broad heterogeneity that has been identified. And we do not know yet what defines this broad heterogeneity. Is it like HLA phenotypes? Is it like disease severity? Is it like age? Is it gender? Is it like ethnicity? So that's everything that we are looking into right now, like what actually defines this broad heterogeneity, what dictates who can still have a good immune response 6 to 8 months after infection. So overall, this is just a summary of like what I've been just telling you. If you look into terms of immunological memory, we can detect immune response for all of these different compartments in almost all of the donors after 1 month and about 90% of 6 months. And it also does not look like this is falling off a cliff. So we're assuming that the immunity may last years, judging from like this first assessment. I pointed out very early that one peculiar thing we have identified is that not only did we recognize T-cell responses in exposed donors, which are shown here in blue, but we also identified donors that recognize these spike-specific or non-spike-specific SARS-CoV-2 specific peptide pools in unexposed donors. So overall, you can see in about 50% of the donors, we identified CD4 T-cell responses, and in about 20%, we identified CD8 T-cell responses. That was very surprising because we knew that these donors have never seen the virus. And the reason we knew that is because we have identified them. We have collected these blood samples 2015 to 2018, so way before this virus was circulating. So when we then went ahead and looked into this in more detail, so what are the epitopes that these donors are recognizing that are derived from SARS-CoV-2 and they have never seen this virus, we performed the exact same study what I've been just showing you before. And that is epitope identification on a granular level. And as I mentioned before, we were very confident that they are unexposed because they have been collected way before the onset of the pandemic. And we have also run IgG analysis serology to confirm that they actually also don't have any antibodies. So overall, we identified 142 epitopes. And again, here you can see the broad heterogeneity of these donors. There's some donors that recognize 2 of these epitopes, some donors more than 30. So overall, around 5 epitopes are recognized for each of these donors. And again, there's some epitopes that are promiscuous that are shared, but the majority of epitopes is unique for each of the donors. So 102 epitopes have been identified in one of these donors. So now that we knew the sequence, we could ask the question, what is it mapping too closely to if you like just look at the sequence homology? So because the knowledge of these epitopes with the exact sequence, we could now like perform blast analysis and very quickly, we realized, okay, it does look like it's mapping to a common cold coronaviruses. So this is viruses that are closely related to SARS-CoV-2 and are circulating widely in the population. And we, of course, then also like synthesize these SARS-CoV-2 homologs for all of the common cold coronaviruses. And if you look into unexposed donors, actually, these are the epitopes that are much more stronger recognized than SARS-CoV-2. So this is just another confirmation that not only do they share sequence homology, but also when you then test them, they are recognized much stronger than the SARS-CoV-2. And as a control, you run these 2 pools in COVID-19 donors, of course, the SARS-CoV-2 epitope pool is recognized stronger than the other pools. So this was like our second hint to not only sequence homology is shared, but also if you then test it for the actual T-cell activity, you see stronger responses. And finally, we then wanted to confirm on a per epitope basis, if you like identify, if you make T-cell lines that are derived from SARS-CoV-2 and then test them against the homologs. So we saw that not only does it also recognize the individual homologs, but many times also stronger. So when you titrate the response, it's much detected in lower concentrations, which means like that they are higher recognized than the SARS-CoV-2. So this was what convinced us then that the reactivity in unexposed donors is actually derived from common cold coronaviruses. I have not shown you that, but like knowing the sequences of both of these subsets is also we do realize it's largely not overlapping. So SARS-CoV-2 does recognize a smaller part that is shared between common cold, but has a huge repertoire that is newly induced. And by that time, we have reported these results. This was also like reproducibly shown in different labs around the world. So these are all the citations that also found reactivity in unexposed donors. So this was something that was like confirmed around the globe in different labs. And that's important because that means like that is phenomenon is also seen in different ethnicities, in different geographical regions. And it's not just something that only happens here in Southern California, but like something that has more like a global application. What was not known at this point, and that's what I'm going to spend the second part of my talk is like, does this have any difference? Does this have any effect? So if you are one of these 50% of the donors that does have pre-existing immunity, does this make a difference when you later get a vaccine, or if you later get naturally infected, or does it make no difference at all? Or does it maybe even hurt you? Maybe you're even like worse off if you have pre-existing immunity. So this is something that we then wanted to ask in our most recent study. And for this study, we wanted to use a very controlled cohort. So we had access to samples from the phase 1 trial of the Moderna vaccine. And I want to point out that we did not test the 100 microgram dose that is currently given, but the 25 microgram dose. And we followed these vaccinees for up to 7 months after initial vaccination. And why we were so interested in the low dose of this was like, because we were thinking that probably if you have a low dose of this vaccine, it's more easier to identify like differences in this pre-existing T-cell immunity. And the other question we wanted to ask is like, how long does the T-cell memory actually last in these vaccinees? Because we had access to 7 months samples after vaccination. So partially, some of this data has been previously reported, and this is all of the part of the antibody data that has been reported. These are the citations where scientists have already looked into antibody responses. So now we were really interested in like, how does that look for T-cell responses? And does this pre-existing T-cell immunity that I have just been showing you have any influence in the outcome of vaccination? This is the overview of the vaccine cohort and the visits we have analyzed. So as I said, we have analyzed donors that have received the Moderna 1273 vaccine, the low dose. So this is a quarter of the dose that these vaccinees have received. They have been vaccinated twice according to their regular schedule, like 4 weeks apart. We have received samples from 35 different individuals. And what I wanted to point out is spanning like all of different age groups, from 18 to 55, from 56 to 70, and also like elderly vaccinees over 70 years old. What we have identified was like different visits. So one was like the enrollment baseline. So this is important because that allows you to look at the T-cell responses before they have received any vaccine. And that is necessary if you are interested in like the effects of pre-existing immunity. The next visit that was collected was 2 days after the first, 2 weeks after the first immunization. Next visit was 2 weeks after the second immunization. And then the final time point was 6 months after the second immunization or 7 months after the first immunization. So this is the visits we've analyzed for all of the vaccinees we have received. And as I pointed out in the beginning, there's two main questions that we wanted to answer. How long does T-cell memory last? And does this pre-existing immunity influence the outcome of vaccination? So the original data I wanted to share you, the first data point is like the antibody responses. And as I pointed out, this has been already like reported by a different group. But also in our hands and with our methods, like we see that there is significant responses compared to day 1 at all 3 time points after vaccination. And the responses are higher at day 43 compared to day 15, and they are strongly maintained up to day 209, which is the 6 months post second dose time point. So this is encouraging. We saw like that 100% of the donors still have antibodies, and that is true for spike-specific IgG, but also RBD IgG in these donors. If you look into neutralizing antibodies, we also saw like that the majority of donors, 88%, were still able to mount neutralizing antibodies up to 7 months after this vaccination. And also similar to like the IgG values. We saw that the responses were higher at day 43, so 2 weeks after the second dose, and then compared to the time points before. And that's important because like we really identified the peak response is induced 2 weeks after second vaccination. So you do see a clear effect of the boost. You do not only see the same response after one dose. So particular for neutralizing antibody titers, you raise the response from 29% of responders to 100% of the responders after the second dose. So what we were most interested is like the T-cell response. And this is just to show you the two different methods that we have been utilizing to study this T-cell response. One is an assay we refer to as AIM, which is the activation-induced molecule markers. That allows you to study the entirety of antigen-specific cells, irregardless if they are able to induce cytokines or not. And that's important because like there's a lot of T-cell subsets, for example, TFH, that not necessarily do induce a lot of cytokines ex vivo in the periphery. So that was one assay that we have been chosen. And this is just for the people that are interested in the details. The panels are plotted here. And the other assay that we have been utilizing was like a more functional analysis, which is the intracellular cytokine assay. And you can see here that these are the different cytokines that we have been studying. So we wanted to make sure that we get a functional profile of these T cells. And we were also able to identify if this is like a Th1 or Th2 or other cytokine functional response that we identify in these vaccines. Quickly to show you here what we've studied. Of course, we were most interested in the spike-specific responses because this is the protein that is used in the vaccine. But because we know a lot of the pre-existing T-cell immunity is derived from non-spike proteins, we are also like measuring the non-spike peptide pools. And we have certain controls, as shown here, cytomegalovirus, because we wanted to see virus-specific responses that are completely unrelated to the vaccine. And then, of course, positive negative control, PGA and DMSO for our assays. So these are the results in terms of the CD4 T-cell responses. And you can see here, CD4 T-cell responses are significantly higher at all 3 time points post-vaccination. And they are again peaking at day 43 compared to day 15. So I also not only antibodies, but also T cells have the peak response 2 weeks after the boost. And interestingly, we saw that very similar to what we know from our studies in natural infection. Also here, about half of the donors show pre-existing spike-specific CD4 T cell memory. And you can see here that the donors group in 2 different subsets, either people that have pre-existing spike-specific immune response or people that do not have that. And that's going to become important later when we look into more detail if that has a difference and if that can influence our subsequent outcomes. So again, importantly, I want to point out that also after vaccination, CD4 T cell responses do not like fall off a cliff, like they are maintained in almost 100% of the donors, 97% have still detectable CD4 T cell responses 7 months after initial vaccination. So that is important because also a low dose of this vaccine induces a very durable T cell response. As a control, I pointed out we measured CMV specific and also the CD4R. So this is like SARS specific peptides that are not contained in the vaccine. And you can see that they do not influence. They are not influenced by the vaccine, neither the CMV specific nor the CD4R, which is the remainder of the SARS-CoV-2 protein, are influenced by SARS-CoV-2 vaccination, spike-specific vaccination. And that's important because there have always been the question if like the vaccine can induce like bystander activation. But from all of what the data we're looking here, it doesn't look like a spike-specific vaccine influences any of the other antigen-specific responses present in these donors. We were then interested in looking into more detail in a subset that is referred to as follicular helper cells, circulating follicular helper cells. And why is that important? Because we know that the CD4 T cell subset is closely correlated with like antibody responses. So if you have a good TFH response, you are able to have like functional antibody formation, class switching of antibody responses that are detected. So this is just to show you the markers that we have been using. We have been looking at CXCR5 positive cells, and this is on day 15 and on day 209. And we saw that in the majority of donors, we do see T follicular helper response induced after vaccination, 75% after the second dose, and we can still detect circulating TFH cells in the circulation 7 months after first dose, 6 months after second dose. So this was encouraging. Also, like functional follicular helper cells were detectable in the majority of donors. So in terms of the functional CD4 T-cell responses, this is the data we have received from the ICS analysis. And you can see here, we detect strong interferon gamma, TNF alpha, T-cell responses, also interleukin 2, and granzyme B responses after vaccination. And if you remember what I've showed you in the panel from our original panel, there's also cytokines that we have been measuring, but we have not strongly detected. So we have not seen interleukin 4 induction. We have not seen a strong interleukin 10 or a strong interleukin 17 induction. So this is why this data is not shown here. So clearly all the functional T-cell responses that was induced was TH1 by, okay, I just heard an echo of my own voice. Okay, so yes, so very functional T-cell response induced in the majority of donors. And as you can see here, again, peak of the functional response, again, 2 weeks after second dose. And we identify functional T-cell responses in almost 100% of the donors after 2 doses of this vaccine. If you then look into like multifunctionality, so that means like instead of like looking at what cells can induce one of these cytokines, is there also cells that can induce 2, 3, 4, or even 5 of the different vaccines, that the different cytokines that we've been studying. And this is what's shown here. So overall, if you gauge on the multifunctionality of spike-specific CD4 T-cell responses, and that is something that is always measured in vaccinology, because that usually gives you a hint that these are multifunctional, polyfunctional cells, and they are associated with like protection against reinfection in other viral diseases. So that's why we were interested in if we also see multifunctional cells. And you can see here, if you follow the response against SARS-CoV-2 spike from day 1, 15, 43, and 209, you do see an evolution of multifunctional T-cell responses. So after the second dose, you have seen the majority of responses. The fraction of multifunctional cells is actually like increasing, versus like if you do the same for CMV specific response, you do not see a change in the multifunctionality subset of CMV specific responses at any time point of the sample studied here. There was always a big controversy if the Moderna vaccine can induce CD8 T-cell responses. So we were also interested in studying that. And you can see here that we do see a strong AIM-induced response in about half of the donors after the second dose. And you can see here that spike-specific CD8 T-cell responses are significantly higher at all 3 time points after vaccination. And 34 donors develop spike-specific CD8 after the first dose, and it goes up to 50% after the second dose. And if you look into further out, 7 months after vaccination, you can see this AIM-specific CD8 T-cell response maintained in 34% of the donors. Also here, we were interested in the multifunctional response. So if you look into the ICS, you can see that in the majority of donors, and that is actually higher than what we see induced in the AIM response, we see a strong induction of interferon gamma, TNF alpha, and also in some donors interleukin 2, only after second dose of vaccination. If you then do the same analysis on multifunctionality, also here you see the induction of multifunctional CD8 T-cell responses following the first, second dose, and also 7 months after vaccination. And again, CMV response is not influenced by this vaccination at all. So this is important because you can see here after 7 months after vaccination, functional T cell response is still present in more than 50% of the T cell responses. And what is also important to point out that this is not necessarily the same donors. So we see in about half of the donors interferon gamma, and in about another third we see AIM responses. So overall, if you like count responses, CD8 specific T cell responses either detected by the AIM assay or detected by the ICS, and together like this is almost like in more than 80% of the donors. And that's what is summarized here. Everything I've been showing you is summarized in this table. So 100% of the donors have strong antibody serological memory after 7 months. 88%, as I showed you, neutralizing titers. CD4 T cell responses are detected in close to 100%. And the CD8 T cell response, depending on what assay you're using, overall in more than 2/3, you can still detect CD8 T cell responses. So overall, we were quite encouraged that even if you get a low dose of the vaccine, you still see a strong immune response 7 months after vaccination. So that was very encouraging because like the responses were readily detectable and also detectable like a long time point after vaccination. So what we were then next interested, okay, so we convinced ourselves that we can readily detect responses after low dose vaccination, but how does this compare to natural infection? So this is what we then were doing next. So you can see here in the red dots, we shown you the response induced by the low dose of the mRNA vaccination, and now directly compare it to what is identified after natural infections, so COVID-19 donors, once they have recovered 6 to 7 months after initial infection. And you can see that there is no difference detectable. And this is true for like spike-specific IgG, RBD-specific IgG, neutralizing titers, no difference in donors that have received a low dose of the vaccine or have been naturally infected with COVID-19 if you look at the 6 to 7 months time point. So that's important because like there has been always the discussion if natural infection might be inducing a stronger response than vaccination. And even here in the low dose, and I want to point out this is 25 microgram, not the one that's given typically right now, which is the 100 microgram dose, we do not see any difference in the antibody level. And also, we do not see any difference in the T cell level. So neither CD4 T cell responses or CD8 T cell responses are different if you compare them to a natural infection. And that's important. That is true for like both of the assays that we have been looking, the AIM assay or also the multifunctional profile, no difference in CD4, CD8 T cell responses if you compare that to natural infection at the same time point 6 to 7 months after vaccination. I pointed out a couple times during this talk that this is not the actual dose that is given right now. So we were interested in like, well, how does this low dose then now compare to the dose that's actually given? And you can see here the data for the antibody responses. And again, in gray here is the 25 microgram, in green is the 100 microgram vaccine dose. And both of these measurements have been done at the peak response, which is 2 weeks post-boost. And you can see here that for all of the antibody parameters measured, for the spike IgG, RBD IgG, but also neutralizing titers, we see a stronger response induced after the higher dose of the vaccine. So this is important because like the low dose looks like natural infection, the higher dose induces stronger antibody responses compared to the low dose. What is the case for CD8 and CD4 T cell responses? Similar to the antibody response, we do see a stronger CD4 T cell, and that is true for measured with the AIM assay or the multifunctional cytokine responses. The response, the spike-specific CD4 T cell response is stronger after the 100 microgram dose. Surprisingly, if we now look at the CD8 T cell responses, we do not see a difference between the 100 microgram and the 25 microgram. So in summary, like antibody parameters and CD4 T cell responses are stronger, and the multifunctional response is higher in 100 microgram vaccinees versus the CD8 T cell response is very comparable between the low and the high dose. So this summarizes the first part of this vaccine study, where we were interested in how long is the response detected after vaccination. And I'm showing you that the low dose, 25 microgram of the Moderna vaccine, induces spike-specific IgG, RBD, and neutralizing antibodies, CD4 and CD8 T-cell responses comparable to natural infection. It does induce lower antibody and CD4 T-cell responses than 100 microgram vaccine dose. And it does induce a comparable magnitude of CD8 T-cell responses than the 100 microgram vaccine dose, although the frequency of the positive donors is higher in the higher vaccine dose. So that means like the response magnitude was not stronger, but more people induced the CD8 T-cell response. So this leaves us with the second question that we were interested in. So now that we know how long the T-cell memory is actually detectable after vaccination, let's go back to the very first question we had is, does this pre-existing T-cell immunity influence the outcome of vaccination? And I already alluded that we've seen in our cohort about half of the people did have a pre-existing immune response. So this means on day 0, before they got the first shot, they already had spike-specific CD4 T cells detectable, versus half of the cohort did not. So this gave the perfect opportunity to now split our cohort into 2 different groups. And this is shown here in this graph. So half of the donors, all shown here in blue, did have pre-existing immunity, CD4 T cells. Half of the donors did not. So the first question we asked, well, we always refer to this as pre-existing immune memory. So is it actually derived from the memory CD4 T cell pool? And this was the case. So these pre-existing spike-specific T cells are mostly derived from the central memory or effector memory T cell pool. So just what we've seen in other unexposed donors, they are derived from the true memory response. So when we now ask the question, is there a difference based on grouping these donors into pre-existing or non-pre-existing immune memory, we can appreciate here that if you look into CD4 T cell responses, at all time points, there's a stronger CD4 T cell response induced in people that have pre-existing immunity compared to people that did not have pre-existing immunity. So that's important because not only after the first dose, but also after the second dose and all the way out to 7 months after infection, after vaccination, we do see an advantage of people that had pre-existing immunity at the time of vaccination. If we then ask if there's also a difference in the multifunctionality of these T-cell responses, and that's what's analyzed here, you can see that at the time point, 2 weeks post first dose, there's a stronger and a faster induction of multifunctional T-cell responses, which then that evens out after the second dose, and also like at the later time point, 7 months post-infection. So clearly, if you have pre-existing immunity, you do induce a faster multifunctional T-cell response after even only 1 dose of this vaccine. So that's a strong difference between people that do not have this pre-existing immunity. Is there a difference in the TFH T-cell responses? So very similar to the multifunctional T-cell response, we also see a faster induction of follicular helper cells in people that had pre-existing immunity, which then again becomes negligible after the second dose or the later time point. But again, faster kinetics, faster induction of TFH responses following just 1 dose of this vaccine. One thing that we are interested in, like if you have a fast induction of TFH responses, does that mean you have a faster induction of antibody responses? And this is the case. So in people that do have the pre-existing immunity at the time of vaccination, they do have a faster onset of TFH T-cell responses, and that also results in a faster, stronger induction of IgG titers after 1 dose of infection. So that is significantly, even if, regardless if you do it with a Mann-Whitney and compare at the same time point or Fisher's exact test. So you have pre-existing immunity, you induce stronger IgG titers after 1 dose of vaccination. The same is true for the RBD-specific IgG titers, which was also significantly stronger induced after 1 dose of vaccination. And what was very interesting to see that all of these faster induction of TFH cells, faster induction of RBD, IgG, and spike-specific IgG titers actually results into like a significantly higher titer of neutralizing titers 7 months after infection. So you can see here, 6 months after the second dose, there's still it's significant, a trend of like higher neutralizing titers in this cohort that had neutralizing titers in the pre-existing immunity in the beginning. So to conclude that, we saw that individuals with pre-existing spike-specific CD4 T cells generate an early multifunctional CD4 T cell responses, more robust follicular helper cells, and faster antibody responses after single vaccination. So overall, I hope I convinced you that we can see a long-lasting T cell memory induced by the Moderna 1273 vaccine. We detect antibodies, neutralizing, and spike-specific IgG durable up to 7 months. We do see multifunctional spike-specific CD4 and CD8 T cell responses for at least 7 months. We do see that this low dose of vaccine induces a response comparable to natural infection, but does have induced a lower response compared to the 100 microgram standard dose. And we also now were able to show that there's actually an effect of this pre-existing T cell immunity after subsequent vaccination. So this pre-existing CD4 T cell memory can influence spike-specific immune responses by the mRNA vaccine. And this is evidenced by greater multifunctional CD4 T cell responses at all time points, robust follicular helper cells, and higher level of spike-specific antibodies after the first dose of vaccination. And with that, I'd just like to point out that this is a true team effort. There's a lot of people involved, not only from my group, but also from, of course, Shane Crotty's group, Alex Tedder's group. And there has been like funding provided by the NIH, La Jolla Institute institutional funds, but also philanthropic support, which really helped us to like get this studies off at an early start in the pandemic. And with that, I like to finish, and I'm ready to answer any questions if I can.
Daniela, that was spectacular. Yay, T cells.
Yes.
Sarada, you want to get us started off in the Q&A?
Absolutely. Again, Daniela, great talk. And just when we're talking about variants, boosters, and breakthrough infections, this is a very timely finding. So thanks for sharing this with us. So a very obvious question to get us all started. This is, you talked about the Moderna vaccine, but would you also extrapolate this result, or would you predict a similar outcome for the other mRNA vaccine, Pfizer, or even the Johnson & Johnson vaccines?
Yeah, that's a great question. And this is exactly what I'm studying right now. So is this true for the Moderna vaccine, or is this also true for other vaccines? So we are right now actually doing exactly that, where we like doing the exact same study after Pfizer vaccination, or also Johnson & Johnson, which are the 3 vaccines that are authorized for use in the United States. So yes, that's a great question, and very much on our mind too. Great. But as of right now, I don't know. Yeah.
Wait, so, Daniela, let me ask you a really stupid follow-up question to that. So how about the like protein subunit based vaccines? So would they be expected to generate the same T cell response, or do you need the nucleic acid template to really drive T cell response?
We know that other subunit vaccines, not for SARS-CoV-2, are very good in inducing T cell response. So I would think that same would be true. We don't know that yet. Of course, we also have access to some of people that have been vaccinated with Novavax. So we hopefully get an answer to this question too. Yes.
All right, back to you, Sarada.
Okay, so 2 questions following up on my first one. So given your findings and given the overall global vaccine shortage, and especially when we're talking about boosters and vaccinating other age groups, should we go ahead and revisit our dosage recommendations because of the lower dose is providing this impressive spectrum of immune response, then why not? Or do you think the lower dose evokes an immune response that is impressive, but then the higher dose would prevent, cases like breakthrough infections?
Yeah, that's a great question, and that's of course on everybody's mind right now as well, given like that a large part of the world has not access to vaccines. So there's two things that I want to point out here. So if this would not be in a pandemic setting, yes, I think you could go ahead and run clinical trials and see like, okay, is the 25 microgram dose just as good in preventing infection or hospitalization than 100 microgram? Given that we are in a pandemic, it's probably not. We probably don't have the luxury of time to do that. So I have been trying to answer this from an illogical question. So does this low dose actually induce an immune response? And we were pleasantly surprised it actually does induce a pretty decent response comparable to natural infection. However, like, is it better like getting any dose into people versus like no dose or like the regular dose? Like that's almost like a question for epidemiologists that can like calculate if that is like better, like we get like 4 times as many people vaccinated with a lower dose, which is clearly not as good as the 100 microgram, but we also don't know the correlative protection, like what is good enough, like to prevent reinfections. So it's a very tricky question. So but certainly like we were encouraged that we do see a durable immune response after like the low dose vaccination. So yes, I'm glad I don't have to make this decision.
All right, Daniela, I've got a lot more questions, but let me remind the audience, please either raise your hand or type your question into the Q&A box and we'll get those prioritized. Until then, Daniela, so, Sarada brought up breakthrough infections. I mean, my guess is it's just way too early to have collected the relevant data, but is anyone working on figuring out, is this more a failure of a B cell response or failure of a T cell response or 50-50?
Yes, so there's a lot of questions. So first of all, we don't know what's wrong under quotation marks in people that have a breakthrough infection. The other part is also like, does this happen after all vaccines or just a specific one? These are all open questions. We don't know that yet for sure, but we are looking into this in particular. So we do have already collected blood from people that did have an infection after vaccination. So they have tested positive after they got the vaccine, and that's something that we are actively looking into right now to answer like, what is different in the people that do have a breakthrough infection. And also one thing I want to point out is like, these people test positive afterwards. Many times they only have mild symptoms. So it's not like the majority of these people have breakthrough infection and then end up in the hospital. So that's the other thing, like that is also like not known yet. Like what is actually like off in these people? Like, do they have a lower antibody response? That's why they test positive, because it allows replication of the virus, or is something different with their B or T cell response. So yes, yeah, I appreciate you asking all the questions of my future work.
So speaking of, there's been some discussion. I saw it early in the pandemic, but I haven't seen any of these results make it to market yet of vaccines that explicitly included T cell epitopes as to try to be an adjuvant. And, there was a big paper from Anna de Groot and Leonard Moise about, predicting best in class T cell epitopes that could be included. Is that in the works? Would you think that's a good idea?
I mean, certainly. So as of right now, we have not seen a variant that is able to like escape the spike specific response. And as I pointed out, it's pretty broad, but like certainly we know that there's a lot of other proteins that induce a very good T cell response. So and I know at least one vaccine that is in development that has like ORF3 next to the spike protein. So I have not studied that, but like that would probably be a good candidate for like, inducing even like stronger T-cell responses.
Mm-hmm.
So, but yes, none of these are licensed, so I don't know yet. It's difficult to compare like what their real life efficacy would look like then, right? So, but yes, definitely a good question.
Okay. So I've got a more personal interest in this next series of questions. I alluded to this in the introduction, but I have a lot of autoimmune disorders, as does my mom, who is also on this seminar. So do you think that what are the implications, both in terms of fighting off the infection and then potential long-term ramifications of, let's say, patient A mounts a mostly B cell driven response and person B mounts a mostly T cell driven response? Who do you think is going to fare better in the short run, and then potential complications of hypersensitivity of those pathways down the line?
Yeah, that's a good question. And that's something that we're just scratching on the surface. I'm involved in like a consortium that does look into exactly like people that either have transplants or autoimmune diseases or a cancer, and see like, is everything that we've shown here, because like I want to point out that what we have studied here is healthy people, right? So is that the same or different in people that have any of these conditions? So we don't know that yet for sure, but that's something that we are actively looking into it, because that is an important question. Like somebody that has an autoimmune disease, somebody that has cancer, somebody that has received a transplant, does the vaccine even induce an immune response, or does it not? So that's something that we are actively working on in this consortium. Taking a step back, I want to point out that it is really important that all of these different parameters of the immune system, the adaptive immune response, have a very different job to do, right? So antibodies are the ones that are the first line in defense. So these are the ones that can prevent infection early on, because they sit in your upper respiratory tract, in your nose. T cells can only recognize infected cells. So that means the virus is already like, circulating. The virus has already like gone inside of some of your cells. So it's possible to think that T cells have a different job like later on in the infection, and possibly could influence disease severity. So that's a question that we don't even know that for healthy people, so let alone the groups that you have been mentioning. So, but that's certainly something that's very interesting and important to find out.
Yeah. I mean, we haven't even mentioned innate response, which is, even well before autoantibody,
yes,
B cells, and, probably is what's saving us all. But, I noticed a report from a couple years ago of seasonal coronavirus 229E having sufficient molecular memory of myelin basic protein that there did seem to be some induction of multiple sclerosis like syndrome from coronavirus 229E infection, and that this was a T cell driven response and not a B cell driven response. I mean, some sort of history, out there to be worried about. And last data point I'll add is that, we're all wondering what long COVID is. I've maintained for, my opinion all along is I think it's autoimmunity. Or at least as a big component of it. And a data point that's becoming more clear in favor of that is that long COVID appears to be much more common in women than men, which mirrors the usual, roughly 9-to-1 incidence of autoimmunity in men versus women, which gets, into X chromosome linked things, and the MHC alleles that preliminary data suggest might predispose you to COVID are all class 1. mostly HLA-B44 and HLA-CO1. So I don't know. I guess I'm just worried.
Yes, so that's a lot of questions you address, a lot of points you brought up here. So
sorry.
First of all, because you mentioned in the immune system, yes, there has been showing that there are certain subset of patients that have antibodies against interferon alpha, like are doing much worse. Like they usually the ones that end up in the not all of them end up in the ICU. So there's certainly like some sort of autoimmune factor in these patients. So we do see that about, I think, in about 10% of the people that end up in the ICU, they do have these autoantibodies.
Yeah, but that's different. That's cause. That's not effect. That's a pre-existing, right.
Yes, yes.
I'm worried about autoantibodies as a result of the infection.
Yeah, well, that's something that we do not study here, but I know some groups are like interested in that. And along these lines, because you mentioned again, this is like the 229E cross-reactive T cells. So that was a question that was in everybody's mind. This preexisting immunity, is this like friend or foe, right? So does it maybe actually like hurt you if you have that before? And that comes from exactly like what you were just mentioning, because we know we do see cross-reactive T-cell responses from all of these 4 coronaviruses. It's not just one. We were interested in like, it's only like one, or like maybe the beta coronaviruses, but it's like all 4 of them. So that was why this was like almost like a very huge relief that is actually like helping you making a better response, because that was in everybody's mind, if that might even hurt you. So, and then I forgot what was the other question.
I don't know. There was too much stuff. Sarada has a question now.
Daniela, slightly switching gears, talking about the pre-existing T-cell response, right? So are you still referring to the pre-existing T-cell response that you explained in the first half of the talk, meaning which is from folks who have not been exposed to this virus at all? Or, we are almost 1.5 years into this pandemic. A lot of us have pre-existing T-cell response from, exposure to this virus. So are you talking about the exposure to the actual SARS virus?
No, so our natural infection studies and also this like vaccine phase 1 trials, they were done early in the pandemic, and they have been running serology and everything, and the virus was not circulating well either in the samples from like collected before 2018 or also in the phase 1 early clinical trials. We do think that this T-cell response is derived from common cold coronavirus exposure. Now, exactly what you pointed out, there is people that might test negative by antibodies, but might have been exposed to SARS-CoV-2 and have actually T-cell responses induced not by common cold coronaviruses, but by SARS-CoV-2 in the absence of seroconversion. So that's something that we are interested in, because we have noticed that there is many times that there is like, couples where like one gets really severe infection and the spouse is always exposed but never seroconverts. So we measuring like, well, does the T cells in these people maybe like induced, or the other scenario where this is like relevant is like healthcare workers, right? Healthcare workers are always exposed to SARS-CoV-2. Not all of them seroconvert, but like we do see some rise in T cell levels. So that's probably an indication of exposure without seroconversion, but like we also don't know what is different in these people that do not respond with antibodies but with T cells only.
Thank you. Do you have any more, Loren?
No. Daniela, thanks so much for your time. Thanks for your research. Keep us posted on these T cells.
Thank you so much for inviting me. Thank you.
Yes, and I hope to see you in person from 6 feet away before.
Yes, close by. Very true. Thank you.
All right. Take care. Bye-bye, everyone.
Bye-bye.
We'll have a next seminar is in 2 weeks. We'll have Jacob Lemieux I forget what he's talking about, but join us again in 2 weeks. Okay, bye-bye, everyone.
Bye.